Hi, everyone, and welcome back to the channel. So in this video, I'm going to talk a bit about cancer genetics. Um, let's start with a bit of a background about cancer. Cancer is a disease of the somatic cells. So we're talking about the cells that make up sort of the normal cells within our body. And unfortunately, about 1 in 3 people will experience a cancer diagnosis at some point in their lives. That doesn't necessarily mean that it's going to be some type of terminal cancer. It could be something as simple as a mole that needs to get removed or something like that. But it could also be a form of metastatic cancer that's potentially life-threatening. In 2025, the World Health Organization expects that about 19.3 million new cases of cancer will be diagnosed worldwide. There will also be 18 million deaths associated with cancer. Now, to be clear, I'm not saying that 19.3 million new cases will be diagnosed and 18 million of those will die. What I'm saying is somewhere in the world, 18 million people will die of some form of cancer that they may have been diagnosed with this year or may have had for several years. In general, cancer is caused by the presence of mutated or abnormally expressed gene products. So we're looking at damage that's been accrued to the genome and then of particular cells. And then this causes changes within the cell that allow that cell to transform into a cancerous cell. And that is the actual proper terminology. The conversion of a normal healthy cell into a cancerous cell is called transformation. Some genes, you can inherit cancerous mutations. Like it is possible to inherit certain forms of cancer due to existing mutations within a particular gene. However, most cancer is going to be the result of spontaneous somatic cell mutations that happen throughout the lifetime of an individual. In other words, it's probably from exposure to some sort of chemical or exogenous mutagen, or it could come from random mutations that happen during the process of DNA replication. Cancer obviously has been well known as a disease for a very long time, and as such, there's been a history of attempting to treat cancer through various means. Right now, the two primary means of treating cancer are, in addition to surgery, obviously, if there's a possibility of removing the cancerous tissue, but usually it is some form of chemotherapy, which may or may not be coupled with radiation therapy. All right. But these are steadily being supplanted by some new emerging technologies and therapeutics to treat cancer. One of them is gene therapy. And another one is there are a series of cancer vaccines that are undergoing clinical research right now with the hopes of actually preventing cancer before it can even begin. Most of these cancer vaccines are actually focused on training a person's immune system to recognize cancerous cells and remove them as if they were some type of virally infected cell, which is really neat because we're harnessing the body's own power, actually enhancing the body's own power to recognize and remove cancer. We should keep in mind that our body already has lots of mechanisms in place to try to prevent this from happening, such as apoptosis, checkpoints within the cell cycle, and also pathways that recognize when there are mutations to our DNA and then correct those mistakes before they can become an issue. Overall, about 5 to 10% of cancers arise from some type of germline mutation that increases susceptibility. So what that means is you could be born with some type of mutation that, while in and of itself doesn't cause cancer, it predisposes you to certain types of cancer. And some of these are fairly well known. BRCA2, for example, is an example of a gene that predisposes you to developing breast cancer. It doesn't mean you will, but it does predispose you to that type of, to developing some form of breast cancer. However, the majority of cancers are still going to rely on some type of mutation, or actually an accumulation of multiple mutations. Usually, mutations that are going to lead to or contribute to cancer are going to impact a few different processes within the cell. They're going to interrupt DNA repair. They might impact cell division through manipulation of the cell cycle. They may block or alter the way in which the cell would undergo apoptosis. So cells that are defective are supposed to remove themselves through programmed cell suicide, which is called apoptosis. So there are lots of cancer mutations that block that process from happening. So the cell might recognize that it's damaged, but then it can't undergo the process of removing itself from the body because this pathway is blocked. It could impact differentiation, so it can make it so a cell doesn't differentiate quite like it's supposed to, or there could be interruptions to cell communication. And one thing that you have to understand is usually, as far as we know, cancer requires multiple mutations. So typically you need mutations in a couple of these different areas to get cancer to happen. You need something that's going to alter the cell cycle so that it promotes cell proliferation growth, right, and growth and reproduction, but you also have to block the apoptotic pathway that would make that cell go, hey, something's wrong, I should remove myself from the population of the body. So usually you need some combination of mutations in multiple pathways to allow cancer to occur. Now, all cancers share two traits. They might come from different origins. They might be in different cell types. They might have different presentations. They might be more or less severe in terms of their effect on the individual, but they all share two common traits. The first is proliferation. So we have to look at abnormal cell growth. Proliferation is the growth of cells and tissues, and then metastasis, which is the leaving of their initial area and spreading to different parts of the body. Now, we can have benign tumors. So benign tumors have the proliferation, but no metastasis. So you may have people say, they found a tumor. And then when they go, oh, it's benign, it's nothing to worry about. What that means is it is a tumor. There is abnormal growth of cells, but it's not spreading beyond that, and it can be easily removed. If we're dealing with a malignant tumor, we're dealing with a tumor that either has or has the possibility of spreading beyond its original confines, which we call malignancy or metastasis. So how do cancers go about? What do the cells in a tumor look like? So one thing we have to recognize is that all cells that are part of a malignant tumor or any secondary tumors that arise from the original tumor are actually clones of the original cancer cell. So what you're looking at is one cell that developed the necessary mutations to become cancerous, and then it's essentially replicated itself over and over and over again, making clones of itself. And this is what that particular tumor, that's what all of the cells in the tumor are going to look like. Now, that doesn't mean that other mutations may develop throughout the progression of the disease. That does happen, but they're all going to share the original set of mutations that led to the cancer in the first place. They may develop subsequent cancers afterwards. And this actually leads to cancer cell subpopulations, which can actually make treatment more confounding. It can make it harder to treat these cancers. You can think of this a bit like the evolution of a cancer, right? You start off with the initial mutations that define it as cancer, and then you can see subsequent mutations occur after the original ones in various tumors throughout the body. For example, I think I skipped, for example, all patients with Burkitt lymphoma will have reciprocal translocations between chromosome 8 and chromosomes 2, 14, or 22. So if you're looking at Burkitt lymphoma, you're going to find this type of mutation at the heart of that cancer. You may find additional cancer mutations that happen after that, but this is what's going to cause the cancer in the first place. All right. A couple other examples when we look at these different types of lymphoma, here's chromosome 14. You can see that if there are translocations involving chromosome 3, it's diffuse large B-cell lymphoma. Burkitt lymphoma is chromosome 8 with 14 in this case. Chromosome 11 can have translocations with chromosome 13 down here, and you can see that 14 with 11, 14 with 18 over here. You can see these all lead to different forms of lymphoma, but they all involve the translocation of this IGH, insulin growth hormone, and place them upstream of certain other cancer-causing genes. And you can see where that translocation happens, and you develop different types of lymphomas based on which gene this IGH gene winds up being adjacent to after the translocation. So there are two different types of mutations that we find in cancer populations. The first set of mutations are what we call the driver mutations. So driver mutations are the subset of mutations that provide that initial growth advantage. These are the things that are going to take that normal cell and turn it into a cancer cell. Usually, this small number is somewhere between 2 and 8. We know it requires at least 2 mutations to cause cancer, but in some cases, it may require as many as 8. So it's this accumulated genetic damage over time that, when combined in the right way, can cause a normal cell to become a cancerous cell, leading to tumorigenesis. Then what can happen after these initial mutations is you can acquire what are referred to as passenger mutations. So these occur in cancer cells, but they don't lead directly to cancer. Okay. This may be the result of increased DNA damage that occurs in cancer cells. We know one of the things that often either leads to cancer or contributes to a cancer phenotype is an inability to either monitor or repair DNA damage. So you can get lots of additional mutations occurring if those cancer cells are not adequately paying attention to the mutations that are occurring. It could provide later advantages if conditions change. So, for example, you might be providing chemotherapy and it's working against the majority of the tumors, but some tumors have passenger mutations that render these slightly more resistant to the chemotherapy, or it could make it resistant to the radiation. Okay. And this can lead into a change in the cancer cell population. So again, you go back to almost like an evolutionary type incident where you're looking at something similar to antibiotic resistance. You're looking at drug resistance, right? The chemotherapy, the radiation, they're going to work against those cancer cells that are the most susceptible to it. But if there are some cancer cells that have these additional passenger mutations that render them more resistant to the chemotherapy, more resistant to the radiation, those cancer cells may survive the initial rounds of these treatments, and then the cancer may seem to have subsided. It may have thought to have gone into remission. They may even think that it's gone because there's so few cells left after that. But if a couple of these mutated cells cling on, they can be sort of like the starting point of that next tumor that comes out. And in that case, that particular version of the cancer might be much more resistant to the treatment that was originally used to treat that, because these are the survivors of that initial round. Again, it's almost as if it's the evolution of cancer at that point. You're supplying a selection pressure and you're choosing for which cells stick around. Which interestingly brings us to the topic of something called cancer stem cells. So the original hypothesis about cancer was that any cell within that tumor could be the basis for a new tumor. So if any of those cells survived, they could then go on and be the foundation for a new tumor. It turns out that that might not be the case. And new research suggests that within the population of tumor cells, there are a small group, well, usually a small group of those cells that are referred to as cancer stem cells. Okay, these are the cells that need to be present for a new tumor to form. So when metastasis happens, it's not sufficient just for any random old cells from that tumor to spread throughout the body. You need to get at least a population of some of those cancer stem cells around, because that will allow the spread of that cancer to new areas or the regrowth of existing tumors. These particular cancer stem cells have been identified in numerous forms of leukemia. This is where it first came from, but now we're detecting them in solid tumors. Now, what's interesting is in some cancers, the population of cancer stem cells may be as low as 1 in 10,000, but in some other tumors, it's been estimated to be as high as 40%. So it really depends on which cancer you're looking at. And what we're not sure of is, where do these come from? Some hypotheses think that these are actually normal adult stem cells that have become cancerous. Others think that there are actually tumor cells that, for one reason or other, take on these stem cell-like properties through the accumulation of mutations. We're still working to suss those details out, but we're pretty comfortable with the hypothesis that within a tumor, within the primary tumor, only some of these metastatic cancer stem cells are the ones that can allow metastasis to occur, which helps to explain why, when you use chemotherapy, or radiation therapy, if some of these cancer stem cells survive because they've inherited some of these passenger mutations, this is what's going to allow for the relapse of the tumor. So there are now new efforts out there to target these cancer stem cells. The thought being, if you eliminate the cancer stem cells, the rest of the tumor is going to begin to degenerate because there's nothing driving the renewal of the cells within that tumor, nor are there stem cells around to be able to propagate that tumor throughout the body. So it's changing the way we're approaching our cancer treatments because we're now recognizing if we don't necessarily have to kill all of the cells. We just have to kill the ones that have the potential to cause the tumor to continue to grow and allow the cancer to spread throughout the body. So another thing that we're beginning to understand is that cancer cell development is a multi-step process. It's not as simple as you get a mutation, you get cancer. It turns out that you get a mutation. Now that mutation is going to be present in every cell that is within the lineage downstream of that cell, right? So green cell gets a mutation, you get all these. Now you need a subsequent mutation in one of these mutated cells, and possibly even more, do you finally get to a cancer cell. These driver mutations, a single driver mutation does not appear to be sufficient to transform a normal cell into a cancer cell. You usually need at least 2, and sometimes, like I said before, as many as 8 different driver mutations to cause a normal cell to transform into a cancerous one. But we have to look at the mathematics behind this because what we're encountering here really is the law of large numbers. Yeah, our bodies have a remarkable set of tools available to them to prevent mutations in the first place. Then if mutations do occur, to repair those mutations. And then if those repairs don't work, to basically remove that cell from either the cell cycle so it can't divide and make new cells that are mutated, or to remove it altogether through apoptosis. But we have to just do some math to look at exactly what our bodies are up against and why it's actually shocking that as little as 1 in 3 people encounter a cancer diagnosis at some point in their life. The spontaneous mutation rate in humans is about 1 in every million nucleotides. So what that means is the spontaneous mutation rate is about 1 per million per gene. All right. So that means, like, that doesn't seem high, right? Like, you only have 25 to 30,000 genes, 1 per million, not high. Well, let's look at the fact that there are going to be about 10 to the 16th cell divisions in your lifetime. So in your lifetime, you will have, if I'm doing my math, that's 10, 12, 15, whatever the above quadrillion, I think it's centillion. You're going to have 10 centillion, if I'm getting that right, or quintillion. I'm not sure what the 5 is. It might be quintillion. So 10 quintillion cell divisions in your lifetime times that number, we still end up with about 10 billion mutations per gene per lifetime. In other words, you're going to get a lot of mutations in your lifetime. You can't help it. You have too many cell divisions, which means that if one mutation alone caused cancer, we would all get it at some point. It would probably be very early on in our life. But because we have all of these different ways of repairing them, because apoptosis is a thing, we have a way of surveilling against these things, and because we require multiple mutations to lead to cancer, that's why we are only at about 1 in 3 people developing cancer at some point in their life. But your cancer risk does increase exponentially with age. Why? Because you're getting through more and more of these cell divisions. Your ability to repair and surveil damage goes down as you get older. Cells start to be a little bit worse at what they do over time as you get older. So all these things contribute to your cancer risk only increasing as you get older. We also know that throughout your lifetime, you are exposed to more and more carcinogens. You just are. It's cumulative damage. If you've ever smoked, if you've ever worked at a chemical plant, if you've ever been in a fire, if you're a firefighter and you breathe in smoke and stuff, you are exposing yourself to carcinogens on a fairly regular basis. We can't help it. We go out in the sun. You go out in the sun because you need vitamin D. Well, those UV rays are also still going to expose you to a potential carcinogen, right? Our own metabolism exposes us to mutations that could potentially cause cancer. So we are bombarded with potentially cancerous, you were exposed to carcinogens on an everyday basis. There's really nothing we can do to prevent it. So all of these facts support the idea that cancer requires an accumulation of multiple mutations, but also supports the fact that our bodies have ways of helping to prevent cancer in the first place. So what are some things that can contribute to cancer? What are some things that we find very commonly in cancerous cells? Well, one of them is genomic instability. So cancer cells often demonstrate high levels of genomic instability. We find lots of chromosomal duplications. We're more prone to find deletions or insertions in there. We're also prone to find translocations. So all of these are kind of the symptom of a cancer cell. For example, chronic myelogenous leukemia, also known as CML, is characterized by a very specific location from chromosome 9 to chromosome 20 to chromosome 22. I have it written as 21, but it's clearly 22 on here. It's called the Philadelphia chromosome, so that's just a typo. I'll have to adjust. It's called the Philadelphia chromosome, where it pushes these two genes, BCR and ABL, right next to each other, and this is what leads to CML. And then I've already showed these examples here, numerous forms of lymphoma arising from translocations between chromosome 14 and chromosomes 3, 8, 11, and 18. So those are some examples. We also find that DNA repair is also often defective in cancer cells. So many cancer cells show mutations that impair the DNA repair process. For example, an inherited form of cancer called xeroderma pigmentosa. This is caused by a mutation in a gene that is essential for nucleotide excision repair. I talk about what NER is in one of my other videos. As a result, they are not able to deal with damage that's associated with exposure to ultraviolet rays. So they often develop skin cancer because that's the tissue that's most prone to being exposed to those UV rays. Hereditary non-polyposis colorectal cancer is caused by mutations that impair mismatch repair. So they accumulate genome-wide mutations, and they seem to be prone to a wide variety of cancers. One of the most common is a form of colorectal cancer. And we talked previously about the BRCA2 mutations. These impair homologous recombination, and as a result, patients have a higher risk of breast cancer. So it shouldn't be shocking to find that cells that have acquired mutations that can lead to cancer are also defective in repairing those mutations. And as a result, these cells are also prone to developing more of these passenger mutations down the line because they're simply not making repairs where they're needed or identifying where repairs are needed. Another very common thing to find in cancer cells are changes to chromatin. So we've talked about the importance of epigenetics, right? Maintaining proper chromatin structure, keeping genes silenced when they shouldn't be expressed, making sure the necessary genes are able to be expressed when they are. One of the things that we happen to observe is altered DNA methylation patterns. In general, we find less methylation than in normal cells, which means more of the genome is open to being expressed. We're not silencing genes that should be silenced in certain locations. What's interesting is we also find hypermethylation in some locations, but the hypermethylation, which silences gene expression, is almost exclusively found in genes that are important for repairing DNA and controlling the cell cycle. So what's happening? We're allowing transcription of genes that can contribute to the development of cancer while silencing those genes that will be responsible for preventing it from happening. We're turning on the gas, we're cutting the brake lines. That's what's happening right here. We've also find that histone writers are aberrantly expressed. So those things that can come in and methylate or acetylate, those writers that we talked about, they seem to be behaving abnormally as well. So you can see right here, just by modifying the chromatin structure, we're rendering cells more prone to developing the cancer phenotype. And not surprisingly, defects in the cell cycle are important. The cell cycle regulates when cells are able to divide. This is typically controlled by a combination of proteins. So you have these enzymes called cyclin-dependent kinases, or CDKs. These are always expressed in the cell, but they aren't active unless they can interact with their respective cyclin. Cyclins go up and down to help control the cell cycle. So when that cyclin is present, that CDK is active and does its job. When that cyclin is absent, the CDK doesn't work and is just sitting in an inactive state. The decision whether a cell should progress through the cell cycle is controlled by both internal and external signals, and then the cells basically put those pieces together and tell the cell what to do. Cell surface receptors record external cues and signal through signal transduction pathways. about whether a cell should be dividing or not. But what ends up happening is a cell that shouldn't be dividing should be pushed out of the cell cycle and into a quiescent state. This is sometimes referred to as G naught, and then they can be brought back into the cell cycle in most cases when they need to divide. So a great example, a cell that is bordered on all sides by other cells shouldn't divide. That should be a signal that that cell, an external signal, it's called contact inhibition. It should be a signal for that cell to say, hey, I'm touching cells on all sides. I'm within an intact tissue. I shouldn't be dividing because there's no space to put the daughter cell when I do divide. But in cases of cancer, the cells that would regulate something like contact inhibition can go away. So mutations in genes that regulate the cell cycle can cause them to sort of ignore those key checkpoints. I have another video out about the cell cycle if you're interested. It's in there with my mitosis and meiosis stuff. And one of the things we talk about is there are a series of checkpoints throughout the cell cycle. For example, there's something called the G1 checkpoint in the cell cycle. That G1 checkpoint occurs before the cell undergoes the synthesis stage of the cell cycle. If a cell is not prepared to divide, if external cues say it shouldn't, if certain internal cues say it shouldn't, the cell should pause at this G1 checkpoint and decide to either fix the situation. So, for example, if there's DNA damage, there's not enough energy in the cell to divide, something's wrong, the cell can pause here and try to solve that problem. But if it can't, or it takes too long to do so, the cell will undergo apoptosis and remove itself because it just recognizes there's something wrong with me. I shouldn't be making more of myself. Or if external cells just say, hey, there's nothing wrong with you, but like we don't need to divide right now because we're in an intact tissue, it can get pushed out into G naught at this point and then re-enter when it's time for it to divide. But if we cause mutations that disrupt the G1 checkpoint, or the G2 checkpoint, or the M checkpoint, a cell that's not supposed to be dividing for one of a myriad reasons can still progress through that. So mutations in genes that are called tumor suppressor genes, genes that would normally prevent cell division under these circumstances, well, if you mutate these in a way that breaks them, a loss of function mutation, these will fail to prevent division. We can also, on the other hand, pump the gas a little bit more. The way this works is by acquiring mutations in proto-oncogenes that basically make them more active. So the way these would work is a mutation in a tumor suppressor gene would likely be a loss of function mutation. You're cutting the brakes. They can't function to stop the cell cycle. Whereas mutations in proto-oncogenes would be gain of function mutations. They would be the gene products are more active to promote increased division. And like I said before, you kind of need one of each of those mutations, right? If you're in a car and you mash the gas pedal, but you also have the brake down, the car still doesn't go anywhere because the brakes are down. So what you have to do is you have to cut the brakes and mash the gas pedal, basically. Mutations in signal transduction pathways that interpret external cues can also be problematic. So for example, if you break the pathway that tells the cell, hey, I'm contacted on all sides by cells, I shouldn't be dividing. If that signal doesn't get to the nucleus, then the cell doesn't know. If those signal transduction pathways are broken in some way, they might go, hey, there's nothing stopping us from dividing. Let's do it. Even though the cell recognizes externally that it shouldn't be dividing. So as you can see, dysregulation of the cell cycle is essential. Without some type of mutation impacting the cell cycle regulation pathways, you're probably not going to get cancer because the cell cycle will kick in and just say, I don't care what mutations you have, we're going to remove you from the cell cycle and you're not going to continue causing problems. And that largely happens through apoptosis, which is programmed cell death. Defective cells in our body and the body of all, multicellular organisms should all have the ability to undergo apoptosis. They can basically say, there's something wrong with me. I'm no longer needed for one reason or another, whatever the case may be. And they undergo this process of programmed cell suicide. It's kind of neat. Signal transduction pathways converge. Usually, interesting, the mitochondria are very important in this process. The nucleus is very important in this process. And the cell ends up producing a bunch of enzymes that are known as caspases. And these caspase enzymes are really good at just digesting all of the cellular components. And these caspases go through and They break open the mitochondria. They release a protein called cytochrome C. This causes the cell to begin this process of cell death, where the cell essentially eats up all the nucleic acids. It eats up all of the proteins. It eats up all the cell components, and it essentially just implodes on itself and disappears, which is awesome, because when a cell undergoes apoptosis, it essentially removes itself for whatever reason, usually because it's prone to damage the entire body. And the cell just kind of implodes on itself, and then it gets eaten by like a macrophage at some point, and now there's no longer a problem. This, by the way, is also how our cells, our bodies get rid of virally infected cells. It induces the same apoptotic cascade, and the cells remove themselves from the body without allowing the virus to spread to other cells in the tissue. So apoptosis is super duper helpful, which is why cancer cells often have mutations in genes that prevent apoptosis from happening. To give you an example, BCL2, and BCL2 is a protein that inhibits apoptosis. BAX is a protein that promotes it. So cancer cells are often found to have BCL2 levels that are higher than normal cells while having lower levels of BAX. Again, we've increased the gas, we've pushed the gas pedal down harder, and we've cut the brakes. The thing that's supposed to the thing that is supposed to promote apoptosis is gone. The thing that's supposed to inhibit apoptosis is now expressed in lower levels, which is bad for the cell. All right, because now that cell that should undergo apoptosis, remove itself from the population, can no longer do so. So we'll talk a bit more about proto-oncogenes and tumor suppressor genes, and I'll give you a few examples of them. So you can see, normal cell cycle, proto-oncogenes are the gas, tumor suppressor genes are the brakes. This is just a picture-based description of what I've been saying to you throughout this video. Proto-oncogenes turn into an oncogene, too much gas, right? That mutation turns it into an oncogene. Tumor suppressor genes get inactivated, we don't have brakes, and all of a sudden now mutations lead to cancer. So proto-oncogene products normally promote cell division. Often these are transcription factors. So remember, that's that small group of proteins that bind to the DNA and regulate gene expression. And the way most proto-oncogenes get converted into a full-blown oncogene is that they're mutated in such a way that their products can't be normally regulated or get expressed at abnormal levels. All right. On the other hand, tumor suppressor gene products are most likely going to be inhibitors of cell division. Quite often, they're regulating cell cycle checkpoints, or they're the proteins like BAX that would normally promote apoptosis when cells are mutated or the DNA is damaged, or growth suppression signals are present. All right. So let's take a look at a few of them. One of the most common oncogenes that's known to be mutated in a whole host of cancers are the RAS gene family. So there are a bunch of different RAS proteins, but the RAS family generally encode transcription factors that signal transduction proteins that trigger cell growth and division. Typically, they're downstream of growth factors. So these get activated when growth factors get released that say, make more cells. It's either time to fill some sort of to repair some type of damage, or the body's got to grow. So this can be downstream of growth hormones that promote overall growth of the individual. But the bottom line is, when they're active, they're supposed to be encouraging cells to grow and divide and make more of themselves. So when we mutate either RAS or other proteins within the RAS gene family or downstream of RAS, what's going to end up happening is we're basically going to mutate them in a way where they get stuck in the on position. So they're constantly telling cells that they need to divide. And not surprisingly then, because this is such a global regulator of cell division, RAS genes are mutated in about 1/3 of all human tumors. And that's not like cancer type specific. RAS gets mutated a lot, and depending on which cell lineage, is it in a cell that's in the breast? Is it in a cell that's in the bone? Is it a cell that's in the skin? Is it in a cell that's in the liver? Wherever that RAS mutation occurs, that cell is now more prone to being cancerous than it originally was. Then we can look over at the opposite. P53 is one of the classic tumor suppressor genes. P53 encodes a protein that is normally found at low levels in the cell, but P53 begins to accumulate in the nucleus when DNA damage is sensed. So if there is chemically induced DNA damage, double-stranded breaks, if there are DNA repair pathways that are active and doing their job, P53 begins to accumulate. as P53 accumulates, that is a signal for the cell to stop. It actually stops the cell at the G1 checkpoint before DNA replication begins and says, we're going to pause right here and we're going to see if we can repair this damage. If that damage cannot be repaired in a timely manner, what ends up happening is P53 will eventually trigger apoptosis. This will lead to the transcriptional activation of Bax genes. And as you recall, Bax is one of those pro-apoptotic set of genes. They're going to promote apoptosis, and we're going to find that that cell is going to remove itself rather than try to replicate and ignore all those breaks in the cell cycle. Not surprisingly, then, we find mutations that inactivate or reduce the activity of P53 in about 50% of human tumors. So you're seeing that's one of the reasons why I bring these two up. They're just very, very common. Now, is a single mutation in P53 enough to cause cancer? It is not. Is a single mutation in RAS enough to cause cancer? No, it's not. Usually, as far as we know, you need at least 2 mutations. Now, if you get a mutation that turns RAS into a full-blown oncogene, and you get a mutation that turns off P53, are you getting cancer? Yeah, most likely, because you've now turned the cell cycle up to max volume, and you've also made it so that there's no way to stop that cell from going through the cell cycle. The last thing we'll talk about are oncoviruses. So oncoviruses are viruses that are able to increase someone's susceptibility to cancer. They in themselves can typically cause cancer. So most oncoviruses are RNA viruses that fall into the category of retroviruses. They have an RNA genome. They use an enzyme called reverse transcriptase that turns the RNA genome back into DNA, but that then allows the DNA to integrate into the host genome. It basically becomes part of that cell's DNA. It's just there now. We now refer to that integrated virus as a provirus. To give an example, in some ways, proviruses can encode U5 and U3 genes that are strong promoters and activate transcription of viral genes. This is often the case with genes, for example, human papillomavirus, the types of HPV that can cause cancer, reproductive tract cancers in women and also in men. And these can typically cause cancer in 3 different ways. Integration near proto-oncogenes. So the U5 and U3 sequences increase gene expression. So if you put this upstream of proto-oncogenes, now you can take these proto-oncogenes and express them at significantly higher levels. This is basically akin to pushing the gas pedal down harder. Why? Because these are encouraging the expression of these proto-oncogenes when they shouldn't be expressed. You can incorporate a host of proto-oncogenes into the viral genome, which can then spread to other host cells through the increased expression of the U5 and those U3 sequences. So what this basically means is if this particular virus brings proto-oncogenes with it as part of the viral genome, now those proto-oncogenes sit inside that individual cell and they begin to express themselves under the control of these U5, U3 sequences. So in this case, it's not actually working on the proto-oncogenes that are in the individual cells. It's bringing proto-oncogenes with it. And as a result, because it's now integrated or part of the virus's genome, as that virus spreads from cell to cell, it brings these proto-oncogenes with it. And that in and of itself can turn all of those cells into potentially cancerous cells at that point. Or it might encode a viral gene that promotes cell division. So there are lots of different oncoviruses that can cause cancer in these ways. We could be talking about HPV, which causes cervical cancer, penile cancers, things like that. We could be talking about hepatitis B, hepatitis C, which increase people's risk for hepatocarcinomas, liver cancers. Even HIV, which is a retrovirus, which can cause, which we know leads to AIDS, also causes various forms of cancer, such as Kaposi's sarcoma. So there are lots of different oncoviruses, and these are predominantly the ways in which they act to cause cancer in the cells in which they infect. All right. So I hope you can see from this video. And you've learned more about where cancer comes from. At the root of it, all cancers are some sort of very deviation of the cell cycle. They're going to cause cells to divide even when they shouldn't, which can eventually lead to proliferation and then metastasis in the case of cancer cells. And they're the result of the accumulated damage of either just normal run-of-the-mill DNA replication, or they can come from outside insults, such as chemical mutagens or exposure to things like ultraviolet rays, or they could be from being exposed to oncoviruses. But overall, this is really a defect in the cell replication process, a defect in the cell reproduction process, I should say, which leads to the overgrowth of cells that shouldn't be growing. So I hope we learned a bit more about the origins of cancer and how that can occur through genetic mutations. I hope you learned a lot, and I'll talk to you all again soon, hopefully. Bye.